Hello, everyone. My name is Ryan Diaz, and today I will be talking with you about forensic identification and metabolomic profiling of two oak species by GC-TOFMS GC. Before I go into any chromatography or chemistry for that matter, let's first look at the global timber industry. Here I've provided a snapshot of it for you, but as you can tell, there's a lot of text and numbers, so I won't be reading this to you, but I will highlight three important pieces. First, globally speaking, wood and wood products are valued at about 244 billion US dollars, or at least they were valued at that in the year 2019. In my adoptive country of Canada, that translates to about $33 billion in export products and almost $12 billion in import products. So, why would the global timber industry care about forensic identification of oak species or any tree species in general? Well, generally speaking, to protect against their profit losses here, right? I mean, this is a big dollar industry here, and nobody wants to pay international fines for illegally sourced lumber. So, our study objectives then are finding some species specific metabolites that do not vary from one individual tree to another of the same species. Another very important question is, can we use a tissue other than heartwood? Although this is the conventionally used sample, it costs quite a bit of money and it also damages the trees. It makes it really hard to maintain sort of a reference material collection. And last but not least, is there maybe one tissue agnostic metabolite panel or different panels for each tissue? Let's find out then, shall we? Of course, to do that, we will be using GC-TOFMS GC-TOFMS, no doubt about it. Let's begin with the study design, right? So we have quite a few different tissues that we're interested in. There are microcores, current year shoots, 1-year-old shoots, foliage or leaves, heartwood, and sapwood. I'll just take this time to note, as you can see here, microcores and sapwood are the same exact tissues. It's just the sample size that differs. And in all of these diagrams, you can see which means what. So, keeping that in mind, we started with a pilot using two oak species, red oak here on the left and bur oak here on the right. These are both economically and ecologically important for Canada, and there are other oak species listed on the protected species list, such as Mongolian oak. We began the study with 5 individuals per species, but we have expanded it since then to be at least 10 individuals from each species. And you'll note here the difference in size of the microcores and sapwood. Now, this is all well and good, so what does this look like in the lab? Well, in the lab, wood samples were ground to a fine powder prior to extraction in acidified methanol overnight, and followed by GC-TOFMS GC TOFMS analysis on a LECO Pegasus 4 quad jet using liquid nitrogen with all of these parameters you see. So now that's in the lab. What do we do on the computer? Well, the acquired peak tables were aligned using stat compare feature in the LECO software. This aligned data was then normalized using the sample's weight and the internal standard response. Data were auto-scaled, and then chemometrics were performed on the data in MATLAB with PLS Toolbox. So, what does this all look like after all of this fun stuff happens? Well, here I will show you some representative chromatograms of both bur oak here on the left and red oak on the right. You'll notice that there are obvious differences just upon visual inspection. That's a good sign. So, this wouldn't be any typical hairnut group presentation without principal component analysis and the scores plot resulting from it. So using all of my samples and all of the variables, I have some scores plots that look like this. On the left, it's broken down by species, with the bur oak labeled in red and, confusingly, the red oak labeled in green. On the right, on this scores plot, everything is broken down by the tissue of the sample. And you'll notice in this region on the scores plots in the bottom right corner, all of these samples are leaves, and that doesn't look very good. So we've concluded leaves are not helpful. They're very bad, actually. They present more compounds in their chromatograms than all of the other tissues that we tested. So chromatograms were then realigned, excluding all leaf tissues, using a signal to noise of 15 and 3 different mass to charge ratios for peak detection. The newly processed samples yielded fewer compounds and cleaner chromatograms, and my new data set now is 109 samples with 8 replicates and 293 variables. So the scores plot resulting from principal component analysis of this data set looks like this. Again, we can kind of see a distinction between our red bur oak on top and our green bur oak here on the bottom. Now, to make this separation even better, and to maybe filter the number of variables that someone like me would have to look through, we can perform our feature selection guided by cluster resolution. This is an in-house algorithm developed by the Hairnut group. There are multiple references on it, but the true detail and nitty-gritty of how this works is far beyond the 10 minutes that I have to talk to you. So it's a big black box, and it's going to help us, reduce the number of variables to help us build a better model. Well, after doing exactly that, we yield a scores plot, something like this, where, oh, look at this. Now we have this red cluster of bur oak, obviously set aside from this green cluster of red oak, and only using 29 different variables. Now, speaking of those variables, these features that were selected, here they are, presented in table form for you. I will not be reading it to you, don't worry. On the bottom here is the PCA is the scores plot that I just showed you after feature selection. And this is the biplot of that same data set. Again, I'm not going to read the analytes or names or anything to you, but the important point I want to note, take a look at the right-hand side of the table. Just about every compound was either present in only one species, or if it was present in both species, there was an obvious difference in the amount therein. So upward arrows represent a compound that was detected in higher amounts on average in that species, and a downwards arrow reflects a compound that was detected in lower amounts on average in that species. So taking these features that were selected, then we could maybe build a classification model. Well, I won't say maybe, I actually did it. So if you perform partial least squares discriminant analysis on the 2 oak species with the data set that I had been showing you before, we will yield a scores plot like this, which is very nice. At this time, I will direct you to our ROC plots over here on the right, and you'll notice that we have 100% correct classification for our green red oak cluster, and 98% classification for our red bur oak cluster. And you'll see right here in the middle of the red oak samples, the green squares, there is one bur oak sample that was misclassified. Now, that could have been a mislabeling or some foolish mistake of my own. We'll find out what in the ensuing days. I'd just like to point out the loadings plot associated with this PLSDA, and you'll note that regardless of which latent variable you pick, all of these variables, all 29, are somehow important for this model. So they are very informative in terms of classifying between both types of oaks. That's a very good sign. So, in conclusion, these wood tissues can be used to differentiate between the 2 oak species. Great. The 29 selected features or variables are important, quote, for classification. I will say now, my PLSDA model needs to be validated. What I mean is that I only had 109 samples, which is a good enough amount for a start, but not a good enough amount to confidently say that I have a working classification model for all possible oak samples that may come my way. So plenty more needs to be done, but this is a very welcome first sign. And last, but certainly not least, this methodology could be applied to other wood and wood products for about the same purposes in terms of forensic identification of them. At this time, I'd like to acknowledge and thank everyone who's given money to either myself or Dr. Harnack. As well, I'd like to thank my group members for their many words of kindness and support, as well as their jives and their jeers.